then did a PhD in zoology from University of Oxford. And then he went on to postdoctoral training, first at Paris, France, and Queen's University in Canada, followed by postdoctoral fellowship at Department of Ecology and Evolution and Behavior at the University of Minnesota. First time I met Daniel when he was a co-director of Biology of Aging course at not far from here at Woods Hole. And so he got his lab in 1995 at the University of Georgia, and he was very successful there and went to the ranks of full professor, and then he was recruited by the Department of Biology and Pathology at the University of Washington. And he is well known in the aging field, and he has a wide range of interests, including systems biology of aging and metabolomics, and he's trying to use these approaches to understand why we age and what we can do about it. And most recently, so most of his work was done in fruit flies, but more recently he started an NIH-funded initiative on studying aging in dogs, and I hope you'll hear about it today. Please welcome Daniel.
Thank you very much for the invitation to visit today. I've had a great time, a lot of really fun conversations in many different areas. The Dog Aging Project is fundamentally an interdisciplinary project, and it's led to me having the opportunity to have lots of interesting conversations with people working in different areas, which is ultimately why we do science in the first place. So it's really been a pleasure to be here, so thank you for the invitation. So what I'd like to tell you about today is systems biology and aging in companion dogs, which is the goal of the Dog Aging Project. We are about to launch what will be the largest long-term longitudinal study of aging in dogs that anyone has ever done. Before I do that, I'm going to tell you a little bit of a background about how I got to this interest, why we're doing it, and then because we're just starting in dogs, I'll motivate it with a few examples of the kinds of work that we do in my lab on flies, and how our fly work kind of inspired what we're trying to do with dogs, although rather than experiments in the lab with flies, most of what we're doing is simply the power of paying attention to people's dogs and not experimenting with them. It's dogs and flies today, but the flies kind of motivate the dog work. But first, a little bit of background. I started working on aging as a graduate student in the late 80s, so I've been thinking about it for a while. And what excites me partly about working on aging is that it's kind of a conceptual hub that links everything. It links biological phenomena, it links biomedical, pathophysiological processes, it even links beyond biology to sociology and economics and philosophy and just any discipline you can imagine. In terms of scale, so we see the effects of aging on diseases. So these are data that I downloaded from the CDC a few years ago. All the major causes of mortality in adult humans: heart disease, cancer, cerebrovascular disease, diabetes, flu, and so on. And it's striking that in every single case of all the major risk factors for aging in adults, the number one risk factor is not smoking or diet or exercise or lack thereof, but rather age. So for all of these major causes of mortality, the doubling time is about 6 to 8 years. These two bold lines represent Alzheimer's disease and Parkinson's, and interestingly, they start late, but they have a doubling time of about 4 years. Aging happens not only across diseases, but also across scales. We can measure the impact of aging from the nanometer scale of how aging affects damage in small molecules, all the way up to many meters, hundreds of meters of large animals like whales and trees like California redwoods. And of course it happens across populations, and the effect varies across populations. Without going into the details, these are data, a map showing patterns of fertility by age and sex from Chris Murray's group at the University of Washington. And this is details of years lived with disability across different wealth categories around the world, from low sociodemographic index to high wealthy sociodemographic index. And you can see in blue a dramatic increase over time from 1990 to 2016 of years lived with chronic non-communicable disease. If you're interested why we see this big increase in these low to middle socioeconomic regions around the world, it's because they're improving over time, and that's leading to more people living longer, and older age people have more years lived with disability. So aging really integrates all these questions. There are really two broad age-related questions that I've spent my whole career working on based on my training in evolutionary biology. And the first is, why is it that there's so much variation across species, from mayflies that as adults live for one day to the longer-lived species, and the longest, the oldest age record for any animal in the world, not that individual, but a picture of that species is on this slide. Which is the oldest individual we know of? Any guesses?
The whale. The whale.
The whale is a good guess, but it only gets to about 200 years. The clam gets to 400 years. And this is the same species that you will eat in Boston clam chowder. So you might pay a special respect to that bowl of clam chowder. There might be a 400-year-old individual that you've just boiled and and then the second big question is about variation within species. So even amongst ourselves and our friends and loved ones, we can see variation in how well people age. Just by what they look like, how they move, their gait, is a pretty good predictor of how long people are going to live. And some people clearly age more quickly than others. Of course, we can measure all kinds of biological phenomena, even just activity levels. So here's some data from a relatively small ski marathon back in 2012, and these are the speeds across the entire, averaged across the whole course. And what we see is a steady decline of about 0.6 kilometers per hour per year of age from the youngest racers in their teens to this oldest racer who was over 80. So I'm interested in understanding this variation. And if we think about a human population, there are two contributors to the phenotypic variation that we see. There's genetic variation and environmental variation, and then the interaction between the two. It's that simple. If we can measure the genetic variation and the environmental variation, we can understand this phenotypic variation. Of course, it turns out to be a little bit more complicated, and we'll get there. So here's one complication. So what we're looking at here is what's called a Manhattan plot. Each one of these points represents a single nucleotide polymorphism in the human genome. And in this case, the researchers have compared lifespan for each one of these points in the different alleles, whether it might be at a particular nucleotide, one half of the population might be A and the other half of the population might be G. And they asked, is there a difference in lifespan? And at this particular gene, ApoE, there's a strong, highly significant effect of ApoE on lifespan. People with the epsilon 2 allele are long-lived. People with the epsilon 4 allele are short-lived. And so that's pretty impressive. And we think, okay, well, we've explained the genetics of lifespan in humans. Of course, as most of you are aware, that the challenge is that while these hits tend to be highly statistically significant, this axis here shows minus a log of p, so a value of 20 means that this has a significance value of 10 to the minus 20. If we look at the proportion of variance explained by the top hits, which is what we're looking at here in this distribution, these are just the proportion of variance explained by the top single nucleotide polymorphism hits for height in blue, Crohn's disease in red, and breast, prostate, and colon cancer in green, you can see that hits like this typically explain between 0.1 and 0.5% of the variance. So if you imagine a hit that explains variation in height, it might be highly statistically significant, but the two groups that are separated by those alleles might differ by a millimeter or even a fraction of a millimeter on average, but if the sample size is large enough, highly significant. So the challenge is to get from genes and environment to, in my case, aging or demography, looking at age-specific survival, age-specific reproduction. And what I'll argue here is that these factors that are statistically strongly correlated with demography, but functionally not so strongly correlated with demography, run through systems biology and physiology. And I'll also suggest that these areas tend to be relatively siloed. Some of us are working on a particular gene and its structure. Some of us are working on a particular omic domain like the metabolome or a particular physiological pathway. Some of us are working on survival or reproduction in the study of aging. And what we really need is an integrated framework that brings all of this together, thinking about those factors in the environment that might have direct effects on demography through extrinsic mortality. And evolutionarily, thinking about how it's actually age structure that ultimately determines how natural selection is going to shape genes, which in turn are going to act through systems biology to shape aging. So my long-term goal is really to develop, work with other people, to collaborate on developing a more integrated way of thinking about aging. There's just another way of saying something similar, but really focused on what we're trying to do in my lab. So lots of genes shape variation in lots of downstream traits. The challenge is that the distance between these two is kind of astronomical. We need a way to bridge that gap, and we can do that through these endophenotypes, the epigenome, transcriptome, proteome, metabolome, microbiome. Of course, the environment plays an important role. In my own lab, we focus on the metabolome, and I'll tell you a little bit more about that. And of course, things like the metabolome can have upstream effects on whether genes are transcribed or not. So it's really a complex network with arrows going in both directions. So why the metabolome? Well, the metabolome is easy to measure in any organism, and it really makes up the fundamental building blocks, both structural, like proteins and phospholipids, polysaccharides, DNA, RNA, and the structural building blocks and the functional building blocks, like the TCA cycle. So if we measure the metabolome, it's kind of a window into the very last step before the traits that we're interested in aging and age-related disease. I'm going to tell you briefly about a few studies that we've done where we are either about to or have already used the metabolome to link genotype to phenotype, and the fly becomes a model for natural genetic variation in human populations. So I'll briefly tell you about 3 studies before I get to the dog aging project. I'll tell you about a new study that we've just kicked off on rapamycin, a study on stress response in flies, and a study on diet restriction. These are all unpublished studies. These 2 are about to be submitted. So the first message is that Drosophila, just like humans, shows genetic variation for everything. And in this case, it's an example of genetic variation for an intervention that can impact aging. And in particular, a drug called rapamycin. Rapamycin is used very commonly in human populations of people who have had transplants. Both solid organ transplant and bone marrow transplant patients are given rapamycin to prevent rejection. It's a molecule that was discovered from soil on Easter Island, also called Rapa Nui, which is why it's called rapamycin. And at low dose, it can increase health lifespan in mice. Here's some data showing mice where rapamycin wasn't started until 20 months, and we still see a significant increase in lifespan. And started earlier in life, we see a significant increase in lifespan. We also see an increase in health span. So for example, mice given just 10 weeks of rapamycin treatment show left ventricular function that looks younger. So it seems to slow aging in heart function. All of these studies are typically done, whether in worms or yeast or mice or flies, in a single genotype. For example, a C57 black 6 mouse. But we are obviously not like a C57 black 6 mouse. We all look really different. So one of the questions we're interested in my lab is whether a drug like rapamycin that seems to increase both lifespan and health span in model organisms would work in a genetically diverse population. So to test this, we give rapamycin to flies that belong to the Drosophila Genome Reference Panel. This is a population of 200 inbred lines of flies that have all been sequenced, so we can ask if the genotypes are different, and if they are, we can map that variation. So here's a study that is just beginning. We have data on development time in flies. So normal development time from egg to pupa in our lab is about 9 days. And here are flies on control food. Here are flies on rapamycin food with a development time that's slowed by about 30%. And here's a line that shows no effect at all. We've surveyed about 80 lines so far, and the mean pupation difference goes from 0 days to about 6 days. This is a 50% increase. So we've got rapamycin resistant lines and rapamycin sensitive lines. We're just kicking off this study, but these are the kinds of data that we can then use to ask about differentiation between the lines. What's special about these lines or these lines? So a couple of examples where we've made some further progress on this. The first is partly to illustrate that the metabolome is a powerful way of kind of magnifying genetic variation. So here are some results where we've measured lifespan in fruit flies that are kept on peroxide food. So this is the same hydrogen peroxide that we collect, that we can buy in the brown bottles in a drugstore. We put that on the food, and it kills the flies, but they die, depending on the genotype, they die on anywhere between an average of about 2 days and 4 to 5 days. So there's tremendous variation, about 100% variation between short-lived and long-lived flies. If we pick a subset of highly sensitive flies that are short-lived and highly resistant flies that are long-lived on rapamycin and look for a genetic signature, it's extremely weak. We can map genes that have some impact, but we certainly can't differentiate a set of, let's say, 8 resistant and 8 sensitive lines. But the pattern is very different if we look at the metabolome. What we're looking at here is a principal component plot. We're looking at the first and the fourth principal component axes for the metabolome. We measured about 3,000 features in the fly metabolome. Each one of these dots represents a sample of 5 flies, which is ground up, extracted, run through a mass spec, and we get measures of all these metabolites. The colors represent 8 different sensitive strains in blue, light or dark blue, and 8 resistant strains in yellow and red. And you can see that they're almost totally separate. So where the genotype data has them completely overlapping, the metabolome data, they're completely separate. Moreover, the light blue flies are on control food, so they've never seen peroxide before we sample. And the yellow flies also are on control food. They've never seen peroxide. And there's almost perfect separation between the sensitive flies on normal food and the resistant flies on normal food. So what this says is that if we measure metabolite levels in flies on normal food, we can predict with greater than 95% accuracy whether that fly is going to be resistant or sensitive if we were to put it on peroxide. So the metabolome is a powerful biomarker for the ability to resist stress. These specific metabolites, maltotriose, maltotetros, metabolites in the starch pathway are strongly associated with this pattern. Moreover, what we see is that in the resistant lines, they don't change from control to peroxide, whereas in the sensitive lines, they do. So the metabolome tells us not only whether a fly is going to be resistant or sensitive, but also points to some mechanisms, starch storage, and also points to this interesting phenomenon that it looks like the resistant flies might be resistant because they actually are able to maintain a metabolome profile that is resilient to any change when stressed by peroxide. So very powerful kind of way of identifying variation that magnifies what was a very weak genetic signature. So the sort of working model is that there are hundreds to thousands of genes that are impacting on the phenotype, but each individual gene has an effect that's too small for us to measure. Many, many genes shape levels of smaller numbers of metabolites, which ultimately shape the phenotype. So if we focus on the metabolite, we can capture all of that genetic variation, which gene by gene is too difficult to capture. So the last fly example I'll give you before I go on to the dogs is to illustrate that the metabolome can be very useful for generating testable mechanistic hypotheses. And in particular, I'm going to talk about diet restriction. I already told you about rapamycin, which is a very powerful pharmacological way of improving health span and lifespan. Diet restriction is probably the most well-known and widely tested intervention for improving health span and lifespan in flies, worms, mice, yeast, and even in non-human primates. And there's some suggestion from the calorie study that people who are in a controlled trial of diet restriction show many biomarkers of improved physiological function. So in this case, we took the Drosophila genome reference panel lines and we raised them, and this was in collaboration with Pankaj Kapahi at the Buck Institute. We raised them either under fully fed conditions or diet restriction conditions. We measured survival for each of these lines, and here's just one example of a line where the red diet-restricted flies are living longer than the control in blue. And then we also measured the metabolome for each line, both under the fully fed, or what we call ad lib, and the diet restriction, or DR, conditions. What you're looking at here are each of those lines and the difference between the diet restriction lifespan and the ad lib lifespan. So if it's above 0 significantly, from about here on, these lines are living significantly longer. There are a few lines that live significantly shorter, as much as 20 days shorter when held on DR food. But we don't know why. We can try and map it genetically, and to jump ahead, when we do that, we don't really find anything. We can also ask whether there are any metabolites that predict this response, and the answer is yes. So first of all, here we're looking at a principal component analysis of the metabolome. The blue dots are all the flies that are held on DR food, the red on ad lib food. So just being in a different food, not surprisingly, has a dramatic effect on the metabolome. But then we also see some interesting patterns. So each one of these dots is a strain, and the line connects it to the same strain held either on ad lib food or DR food. So you can see for threonine, for example, in every single genotype, there's a significant decline in the levels on DR food. For inosine, there's a significant increase in almost every line on DR food. For alpha-ketoglutarate, there's this interesting highly significant interaction term. There are some genotypes that show a decline. There are some genotypes that show an increase. And I don't have time to tell you more about this other than to say that it turns out that if you have high levels of alpha-ketoglutarate, you tend to be more responsive, positively responsive, living longer, to diet restriction. I told you that there's almost no genetic signature, but there's a strong metabolomic signature. And here's sort of the test case for what I was suggesting earlier, that the metabolome can be a kind of bridge between genotype and phenotype. So let me walk you through this a little bit. So the gray circle represents positive response to diet restriction, living longer under diet restriction. And some genotypes do, some don't. We can predict whether a genotype is going to live longer or not based on levels of certain metabolites And so you can see there are about 20 metabolites here, and there are about 20 metabolites here that are significantly predictive of whether a strain is going to live longer or not. So here we see, for example, phenylalanine, leucine, serine, these amino acids predict whether flies are going to, measured under diet restriction, predict whether flies are going to live longer under diet restriction. None of these genes are significantly associated with lifespan, but many are predictive of metabolite levels. This, for example, CCHA2R, which is a gene that's associated with sensing nutrients, interestingly, is significantly associated with a whole set of metabolites, with the majority of metabolites that are in turn associated with the response to diet restriction. Again, I don't have time to go into the details, but we've done validation where we knock out CCHA2R and we show that that gene actually is associated with the response of a fly in terms of its lifespan to diet restriction. So that the metabolome is a very powerful means of linking genotype to phenotype. So these are the sorts of things that we're doing in my lab to try and better explain phenotypic variation for aging and age-related traits that we care about. And so in that sense, I would suggest that these flies can be a very powerful translational model for understanding genetic variation in humans. But what I'd like to suggest is that there's an even better model, and that's Frisbee here, who's my dog, and all the individuals in this species, which represent a highly genetically variable species, a highly phenotypically variable species, and an opportunity to generate results that might inform our understanding of humans and allow us to actually test back in the lab in flies. So how did I get interested in dogs? Having worked for most of my career on flies, this paper came out in 2007 from Elaine Ostrander's group on body size in dogs. So here's a Chihuahua, here's a Great Dane. There's more than 50-fold variation in size between the smallest and the largest dogs. And in fact, there's more than 50-fold variation in risk of many diseases between one breed and another breed. Tremendous variation. Two things piqued my interest, though, and made me actually want to work on dogs. So the first is that a single gene, insulin growth factor 1, was the best single predictor of size. And I told you already that in genome-wide association studies, most genes that are significant statistically are not very biologically significant. In this case, this gene explained about 50% of the variation in size. I'll show you a figure in a few slides. IGF-1 has been associated with aging in lab strains of mice and even in humans. So that got me wondering whether this size allele might have something to do with variation in lifespan among dogs. And that leads me to the second part of the story. So while I was a graduate student at Oxford, I did my research on comparative studies of aging in mammals. So asking why it is that whales and elephants are long-lived and mice and bulls are short-lived. And the first factor, of course, is size. Here we're looking at longevity versus size in both birds and mammals, and larger species are longer lived than smaller species. That tends to be true in general, with some really cool exceptions like bats and naked mole rats. In dogs, it's the opposite. Large breed dogs, like Great Danes, are much shorter lived than small breed dogs like Dachshunds and Chihuahuas. So there's something interesting going on. So that got me really, piqued my interest, got me thinking that dogs might be a great model to figure out something about aging and why it is that these guys are so much shorter lived than these guys, and whether it has something to do with IGF-1. We still don't know whether it has anything to do with IGF-1, but it got a whole group of us eventually excited about working on dogs, and that led to what I'm here to talk with you about today, which is a large national study about to kick off, the Dog Aging Project. So why are dogs a good model? Well, there's huge genetic variation and phenotypic variation. not only in size, but also in behavior. So dramatic behavioral variation. Here's a collie that's busy herding some sheep, and that is innate behavior. There are dogs that love to jump. There are dogs that like to dig. Unfortunately, mine is one of those. And as I already told you, there are dogs that age very quickly, and then dogs that take a long time to age. This genetic variation has already been used to study important diseases. So here's an example of a paper looking at two different malignancies, hemangiosarcoma and B-cell lymphoma in golden retrievers. So very powerful model for mapping disease traits. So I told you that genes and environment are what explain all variation. So we know that genes are important in dogs. People have already been using genes to explain variation. There's lots of environmental diversity as well. So there are about 60 million households, almost 100 million dogs that we know of in the US, in all kinds of environments. It's a little bit hard to see, but this is a dog house, and that's a dog in front of its dog house, and this is home for other dogs in this country. Here's a dog that's been hiking, here's a dog that's been smoking. There are rich dogs and poor dogs, there are healthy dogs and unhealthy dogs. Dogs are found in homes of all racial and ethnic backgrounds, and dogs are found across homes of all socioeconomic status, from very poor to very rich. So tremendous environmental variation. And most importantly, a key motivator for the Dog Aging Project is that people love dogs. Nobody ever comes up to me after a talk on fruit flies and says, "Oh my goodness, I love flies." So for the rest of my time today, I'm going to tell you a little bit about some of what we've already learned from studies based on existing data sets, and then I'll tell you about what's about to happen. We're about a month away from launch, and this is going to be a very big project, and it's going to keep me and a whole bunch of other people busy for the rest of our careers. So first of all, just the basics of morbidity and mortality in dogs. And here's just a little guy saying, "No, that's just the problem, doctor. I'm not a Dalmatian." There is tremendous variation, not only in what dogs look like, but also in the diseases that they get. So from the veterinary medical database, we were able to collect over 80,000 data from 80,000 dogs with help from my colleague Steve Ostad. Each dog had a designation of breed, age, sex, and a description of its diagnosis at a terminal visit in a veterinary teaching hospital. There are about 30 in North America. And so here's an example of an Akita in the 4 to 7 year age range. It's female, it's spayed. In veterinary science, there are 4 sexes, male, female, intact, and sterilized. And here are the diagnoses that had at death. Working with Dr. Kate Creevy at the University of Georgia, where I was at the time, we collaborated. She and her fellow diagnosed a specific cause of death for each dog where it was possible. Often it just said death due to euthanasia. Of course, those had to be removed, uninformative. And we learned all kinds of things. One of the things that we were able to do is just categorize the kinds of diseases that occur in dogs and how those change with age. And for common dogs, that wasn't new to the veterinary world, but for rare dogs, it was actually quite helpful. Here's just an example of 4 breeds showing the kind of variation for the percent dying from respiratory disease, cardiovascular disease, hematological disease, and GI disease. The bulldog, as many of you know, has serious respiratory problems. This is common among the brachycephalic or flat-faced dogs. In a Chihuahua, we see heart failure. Retrievers of all kind get leukemia and lymphoma. And then this interesting guy, this is a Shar-Pei. Its coat is wrinkly due to high levels of hyaluronan, and it suffers from skin problems and also from GI problems, which we think are probably due to those high levels of hyaluronan. So the spectrum of diseases in each breed is very different. What I'm showing you here is for each age, the proportion of individuals that died of a particular cause. So of all those individuals dying in the first year of life, for example, how many are due to cancer? And what we see in humans is an increase and decline. In dogs, we see an increase and decline. Ingestion of toxins, drugs and poisons starts high and goes down. In humans, starts high and goes down in dogs. But dogs don't always look like humans. So for cardiovascular death, with age, by the end of life, after our 80s, it's the single most common cause of death. Cardiovascular death is disease in dogs is relatively low and stays relatively low throughout age. We can look at comorbidity in dogs. What we're looking at here is life expectancy in humans at 67 years as a function of chronic conditions. People with 10 or more chronic conditions are unlikely to make it to 70. People with none are likely to make it to 90. Dogs turn out to be a great model for comorbidity, and this is something that the National Institute on Aging really cares about, because we want to know, it's clear that comorbidities increase with age. We don't know to what extent aging causes comorbidity and to what extent comorbidity increases risk of aging. This is just to show overall dogs have a comorbidity in terms of number of diagnoses of about 3 in this data set from England that we looked at. There are certain conditions like obesity that are associated with increased comorbidities. I'm actually really excited about the opportunity to use dogs as a model, looking at how obesity affects aging and disease. I've talked to many dog owners who will say, well, you know, I can't say no to my dog. I give her treats all the time. She's a lot overweight, but it's okay because she's a dog. And I think our data are going to suggest that it's actually not okay for dogs to be obese, that they suffer because of it. We can also think about comorbidity networks. What are the diseases that are associated with each other? And because of the relatively, unfortunately, short lifespan in dogs, we have an opportunity to ask about the causes of these interactions. And ultimately, my hope is that we can actually map, if we can recruit enough dogs, that we'll have the power to genetically map the genes that underlie these edges linking the various nodes of disease. And this is work from a graduate student in my lab, Kelly Jin, and interestingly, some of the relationships are unsurprising and positive, like pancreatitis and diabetes, but there are also some negative correlations, for example, between epilepsy and stomach torsion. We have no idea why, but it's an interesting hypothesis to explore. As I mentioned, there are 4 sexes in dogs because they're either male or female, sterilized or intact, and we can ask how that affects disease and aging. And this is work that a former graduate student from my lab, Jessica Hoffman, did. Really beautiful study showing a strong effect of sterilizations. And sterilized dogs tend to live longer. This is true for both females, shown in red, and males, shown in blue, that there's an increase of about a year or more. This is from the American Veterinary Medical Database. Here are data from the UK, Vet Compass, again showing a significant increase for females and males who are sterilized. What's really, the one caveat here is that these are cross-sectional studies and not longitudinal studies. We don't know when these dogs were sterilized, and we don't know what their parity was, whether they reproduced or not, before they were sterilized. So this is a question that we'll be able to ask in the dog aging project where we follow dogs longitudinally. What's really interesting to me is that we also ask what these dogs die of. So what we're looking at here is the relative risk. So bars above the line are for causes of mortality that increase after spay-neuter. So dogs that are spayed or neutered have higher incidence of cancer and autoimmune disease at death. The width of the bar is the proportion of all dogs that die of that disease. So you can see that the most common cause of death in dogs is cancer. Also common in dogs are death due to trauma and infectious disease. These are reduced after spay-neuter. And this is not just an effect of age. So cancer increases with age. Dogs that are spayed or neutered live longer. That's not why cancer is greater in spayed and neutered dogs. Even after we control for age, looking at young dogs, we still see that impact. We don't know what the cause of this is, but it's a pretty intriguing result. We've also begun some pilot studies looking at the effects of rapamycin in dogs. I told you about rapamycin in flies, in mice. I told you that it improves heart function. So we've started some studies looking at the effects of rapamycin in dogs on heart function. And when I say we, I really mean my colleague, Matt Kaberlein, who's taking the lead on this part of the dog aging project. So the phase 1 study, and I'll just show you one result, it's a placebo-controlled double-blind study. Dogs are given 10 weeks of treatment. In this case, the primary outcome was adverse events. We were looking to see that it was safe, and we did see no adverse events. It appears to be safe. And dogs were given an echocardiogram at 0 weeks and 11 weeks. So here's a measure of fractional shortening, measure of left ventricular function, and the treated dogs are in red, and the placebo dogs are in blue. There's a marginally significant improvement that dogs on rapamycin appear to have improved fractional shortening. This is just another way of showing that. It's a weak effect, but importantly, things didn't get worse and they didn't have other adverse effects. We've just completed a phase 2 study in College Station, Texas, where our lead veterinarian is, and again, we saw no adverse events, so we're pleased about that. We're working closely with Eleanor Carlson on the genetic side of things. Eleanor leads a citizen science project called Darwin's Ark. She's based here at the Broad Institute, and she's especially interested in the genetics of behavior. So if you have a dog, you can go to Darwin's Ark, you can sign up your dog, and it doesn't cost you anything. You can answer a bunch of questions about your dog's behavior. She'll send you a cheek swab. You swab your dog's cheek, put it in the mail. It will go to GenCove in New York for sequencing, and they will then map traits. And I'll just show you one example. So the survey looks something like this. Very simple questions like Jenny enjoys life. We did this for Frisbee, who I already showed you a picture of, and my daughter filled out the survey. My daughter's pretty smart. She's an MD PhD student here in Boston, but she didn't make it through the entire survey because she got to the question, your dog thinks it's a human, and she didn't know what to say, so she quit. And I think Eleanor has taken that question off the survey. All the questions are on a Likert scale from strongly agree to strongly disagree. It's a very simple survey. And I'll show you one result. Question, or the statement is, dog is quick to recover after being startled or frightened. And 885 dogs have both answered the question and had their cheek swabs sequenced. And so they've analyzed that, and they got a pretty nice hit with the top hit was almost 10 to the minus 9 p-value, highly significant. And the hit is at a gene that's orthologous, a gene in dogs, and sorry, this is a Manhattan plot with the dogs showing 39, so dogs have 39 chromosomes, minus log p. So the hit is orthologous with BRK2, and this is interesting because it's associated with schizophrenia in humans. And Eleanor tells me that one of the prognostic indicators predictive indicators for schizophrenia in humans is the inability to recover after being startled. So potentially interesting connection. So we've already established that even for relatively complex traits, we can use citizen science to obtain data and map these traits. So all of this leads to the last part of my talk, where I'm just going to tell you about what we're actually doing, the dog aging project. This is a national study of aging in dogs to really complete this equation of explaining phenotypic variation in terms of genes and environment and the interaction between the two through systems biology, in particular through the epigenome, the metabolome, and the microbiome. This is just to say that we have a big team of people working on this. There are 4 projects that I'll tell you about. There are 4 cores to support those 4 projects. There are people, collaborators around the country, and we have companies that are on board helping us with managing much of the data. It's really inspired by, especially by the first American long-term longitudinal study, the Framingham Heart Study, and others. A dog long-term longitudinal study, the Golden Retriever Lifetime Study, that's about halfway through its project. And then more recently, these long-term studies that are incorporating precision medicine, genomics, and other omic measures. So we now have a website. You can go there and nominate your dog. The initial survey takes 2 minutes, and you will tell us, if you nominate your dog, its name, its breed, its age, its sex, and where you live, where the dog sleeps, and whether you take it to the veterinarian or not. Having nominated your dog, once we launch, which is going to be in November, we will then send what I hope will be more than 100,000 people who nominate their dog, a large 200-question survey that takes a couple hours to fill out, that is the health and life experience survey, all about the dog and its environment, its health, history, and there will be an opportunity to upload electronic medical records as well. So this is all funded by a U19 from NIA. We will be enrolling 10,000 dogs in 3 named cohorts supported by this U19. I'm hoping to enroll at least 50,000 dogs to complete that 200-question survey. Everybody who completes that survey will get a certificate, and they will be told that they are in the Dog Aging Project Pack, the DAP Pack. And my hope is that we will continue to follow these people using social media to retain them for the rest of their dogs' lives, collecting valuable scientific data. So the step 1, I told you about the nomination survey. The step 2 is the health and life experience survey, and then uploading of the veterinary EMRs. Among those individuals that upload the EMRs, that get to that step, they are then eligible to be chosen for one of 3 cohorts. There's the foundation cohort. All those dogs are going to have gene sequencing and electronic medical records. Within that is a precision cohort of 1,000 dogs. Those will have metabolome, epigenome, microbiome. They'll all have activity monitors on their collars. We'll collect, for all the dogs, we'll collect environmental data, air quality, like particulate matter, carbon monoxide levels, sulfur dioxide, and so on. Air quality, green space around the neighborhood, the distance to the nearest dog park, time spent outside, all those sorts of things. And then about 500 dogs, so 5% of the Dog Aging Project, will be dogs enrolled in this intervention cohort of the rapamycin study. It's a double-blind, placebo-controlled study. These dogs will all be dogs that live near one of 7 veterinary teaching hospitals where their owners will take them for echocardiograms, twice yearly exams. And they'll be on RAPA for 3 years or on placebo. These will be supported not only by our external independent animal welfare advisory board, but also by a data safety monitoring board. So we're treating it just like a human clinical trial. I won't go through all the details. This is the logistics. So we have a recruitment and retention core that gets owners to sends owners kits, which they will then bring into the veterinarian, sent to the reference lab, processed, or sent to a biobank at Cornell University. And ultimately, all the data, whether it's gene sequencing, metabolomics, surveys from the owners, all of these data, environment data that are publicly available, end up at the Broad Institute on their Terra platform, which speaks to the Google Cloud. So all the data will be in the Google Cloud, accessible by everybody in the world through the Terra platform, and with the proviso that anything we need to do to protect the privacy of the owners, we will do. So no private data will be made available, but all the publicly available data will be housed at the Broad, and our goal is that everybody will do science with us. So just quickly, there are 4 projects. The first one is really creating geriatrics for dogs. There are about 40 veterinary specialties, and geriatrics is not one of them. So a veterinarian doesn't have a framework to say whether a particular dog of a particular size and sex that comes in to the clinic is a healthy ager or not. Project 1 will give the veterinary community those metrics and will define how well each dog in our program is aging. Project 2 is the genetics that will try and identify the genetic determinants of that healthy aging. Project 3 is the systems biology, and then Project 4 is the triad trial that I mentioned. But in addition, we hope that other people will write ancillary proposals to fund other studies, that we are creating a framework that will allow people to do all kinds of other things. And not only in science, right before the seminar, I had a terrific meeting with Catherine Wang about the possibility of using the Dog Aging Project to understand better how we communicate genetic findings to the public and how they interpret the meaning of risk. So there's just a limitless possibility of things that we can do, and so the goal is that other people will write proposals to fund other studies using the Dog Aging Project. So partnership with the scientific community. We are formally partnered with the Baltimore Longitudinal Study of Aging with Luigi Ferrucci. We're currently designing a survey for that population to ask them about dog ownership. We're also excited about the idea of potentially having matched cohorts of dogs with dog owners in studies like All of Us, and we're also excited about the opportunities for educational outreach. So our formal launch will be November 14th. Yesterday I had a really great time doing an interview at WBUR with Here and Now. So that will be coming out on November 14th, so big launch. We're in quiet mode right now. We've had about 4,000 dogs sign on to that initial portal. My aim is 100,000, and I'm sure that we will get there. This is just us being in quiet mode. A couple of results so far. The most common dogs are Lab Retrievers, Golden Retrievers, and Shepherds. And surprisingly, the most common name is Daisy. We have a wide distribution of ages, all the way up to an impressive 2 dogs that are 24 years old. Sizes from less than 10, lots of dogs less than 10 pounds, lots of dogs more than 100 pounds, and dogs in every state in the country. So I hope those of you who are interested will join us on this project. If you do molecular biology, we will have a biobank, and we want it to be a biobank with deposits and withdrawals and not a biohoard. So I welcome your participation there. And just to finish up, I'd like to acknowledge the many people who have been involved in this, and especially the folks at NIA, Felipe Sierra, Francesca Macchiarini, Ron Kowalski, and Luigi Ferrucci, who have really been partners in helping make all of this possible. And thank you for your attention.